Okay, antibody structure and function. We'll talk about properties of antibodies, methods of isolation and characterization of immunoglobulins. I'll take a little bit of, in a short pattern today. We'll talk about types of antibody, structure and biological properties, and the immunoglobulin superfamily. So this is one of the most important lectures that you have in immunology. And again, for concept forming that will base, for example, you wonder what's happening in glucose test, what's happening in pregnancy test, what's happening in blood group test, what's happening in step throat test. All these different immunological tests are based upon antibody information. And as I said, I'll give you a beginning. The information is too intense, and a lot of content matter, and I will also help you. I'm not going to tell you when did I graduate from the medical school, but it's been a while. And I remember first year, second year, we used to prepare mnemonics, and I'm going to give you some of the mnemonics. And the extra credit is that each and every pair should make a mnemonic for today's lecture. And I'll give you my mnemonic as well. Mnemonic means something that aids to your memory. Because I don't want you to remember it for first lecture. You really remember it for good. You have to have very good retention. I'm going to give you weird kind of analogies and examples. You just have to bear with me. Okay? Okay? So today, I'm going to start with one of the analogies. Those of you who were old enough to watch this movie, The Incredibles. And this quotation is from the writer and creator of Incredible. And you can see there's a good reason. And remember that my mom and dad analogy of B cell and T cell. And interesting. So dad is always expected in the family to be strong, so I made him strong. That's what he said. And I'm going to do that in today's lecture or immune response as well. Moms are always pulled in a million different directions. You agree with that? Okay, there you go. And now, just imagine lymph node as a home. That's where immune response is taking place. I'm going to give you cells, their function. What your assignment is, based upon this kind of an idea in Incredibles, each and every character, so you can see there's tons of character, I'm going to go in detail. Each of one have very specific purpose. And if you remember, if you can characterize those cells that are here, that will help you to retain the information that I'm going to talk about. And there are tons of them. So I want you to make a character of each and every cell, like a T cell or B cell or NK cell. So these are the players. Remember, they are living in a home. They are living in a lymph node. There's a mom and dad, kids, and everything. So right from a knock at the door, where an antigen wants to come in, right? So there's a peephole over there. You want to see who is this. You want to recognize that. There's going to be doorbell. And there are a lot of different mechanisms that we have today. So keep that in mind. That will help you to remember what I'm going to talk about that. Okay? And I would want you to read it up. It's there on Google. But the creator of Incredible basically really deserves a credit. Because something that if you watch that, is going to stay in your mind for good. And of course, you have to memorize, you have to cram. There's a movie as well, I'm not going to play that. Incredible kind of a movie. So anyway, when an antigen knocks at your door, so what's the very first thing you would do? You want to identify that. You want to recognize that. Whether this is a known fellow or unknown fellow. And the questions then come into your mind. You look at the peephole and see who is this. And then again, if it's a policeman, of course, it's going to dress like a policeman, so on and so forth. And if it's not a stranger, you're going to ask for an ID. I'm pretty sure if you have grown-up kids, you will train them how to answer doorbell. And then again will be timing as well. Right? This is the time of such and such person. So to cut the story short, anything that our immune system sees, we call antigen. The antigen has the capability to be seen, recognized, and processed by our immune system. And anything that our immune system wants to respond to and get all the act together, we'll call it an immunogen. So it kind of stimulates the whole immune response. So by the end of the day, 40, 50, 60 years ago, they knew when you get infected, when something happens in your body, there is a change in your blood. There's something in the blood. Something in the blood. They want to figure it out. For example, you have sugar, there's some information in the blood. If you are pregnant, there's some information in terms of blood. If you have a strep throat, so what you're trying to do is that in all diagnostic tests, you either are looking for an antigen or you're looking for an anti-serum. That was a term that we used to use many years ago, but now we call it antibody. So antigen, a response, and antibody. We talked about antigen, we talked about the characteristics of antigen. So today we'll talk about antibody. So antibody, again, they are soluble proteins. And when they ran that, they took the protein out and they ran it on electrophoresis and want to see what is the pattern of protein. Because of course there are different types of proteins, and they figured out that the antibodies are soluble proteins. And they are a response to our antigen or immunogen. And there are two types of antibody. One antibody is, we call it secreted antibody, that's secreted. The other antibody, we call it bound. There are two types of antibody. And when we do the gel electrophoresis, you see a pattern there. There's a pattern. That's where the names are going to come from. And then again, this is a globulin family. If you remember your blood physiology, there's an albumin there, there's a globulin there. There are many things there, many proteins there. So one of the protein is globulin, and more specifically is immunoglobulin. So that's why it's called IG, immunoglobulin. Two important features of antibodies. Number one, they are very specific. Remember, one of the hallmark of immune system is that it's very specific. It is not random. If it is non-specific, that's what innate immune response is. When it becomes adaptive immune response, acquired immune response, two most important features shared by antibody is specificity. So specificity is basically a characteristic, but it is defined in the structure of antibody that I'm going to talk about today. The other important thing is that it has a biological activity. It's a biological activity. Just imagine a stranger knocks the door, and one of the kids goes over there and opens the door, and that happens to be an antigen, a foreign material, immunogen. It wants to invade. It wants to take over. It wants to cause destruction. So you need to conquer that. You have to overpower that. So that overpowering and conquering or soothing it down is one of the properties of antibodies, and that's what we call biological activity. And there are many processes that this body would do that. And remember, this is all based upon, it's basically you open the door and let the antigen come in or immunogen come in, so you know basically are going to physically handle that. Because if you don't open the door, you just recognize it, close it, we are okay. But once you want to open the door yourself or antigen causes it to open, then you have to restrict that invader. Right? So you're going to hold it, do whatever you want to do. But in biological term, we want to neutralize the capability of invader. We want to immobilize that. We want to agglutinate that. Agglutination will mean if there is one invader, a bunch of people over there, they'll all kind of go and overpower that. Or we want to precipitate. So these are like chemical reactions that we normally do, and I'll take them in detail as I move along. Now, when you take the blood out and let it clot, so the serum is left. That's where antibodies are. So this means this is not cell. This is a cellular part. That's what the proteins are. And if you run on electrophoresis, you come up with at least 5 different components. And based upon the size of those immunoglobulin, we either have an alpha, beta, gamma, so on and so forth. So at least 5 of them I'm going to talk about: gamma, alpha, micro, delta, and epsilon. So these are 5 important antibodies that I'm going to discuss today. And historically, what happened was that, remember when things go out of control, like in cancer patient, one of the cancers is multiple myeloma. So in this cancer, when we run the serum of the blood, that's what run by one of the British physicians 50 years ago, and he found out that there is a special pattern. Let me show it that to you. So what he did was he ran that serum of the patient, right? And you can see red one and a normal control. So if you let proteins go from positive to negative terminal, you will see albumin stands out because that's a lot of albumin over there. And there are a bunch of other proteins, alpha 1, alpha 2, beta. And then he noticed that one of them is there as a gamma, and this happens to be a myeloma protein. So what is happening in this cancer is your body should have produced antibodies in a very coordinated, orchestrated way, but it gets out of control. It makes too much of a product, which is myeloma antibody, and that leads to cancer. Okay? That's why we need to genetically control that. But you can see from here, that's how we figured out what the pattern of mobility is, because it's just like you make 5 people run, the chances are the heaviest one will be little, but it will slow down. So on electrophoresis, the heavier will not run fast. So the lighter you are, the faster you're going to be. Okay, the other important thing is that we are going to go in detail the structure of antibody. That's a biochemistry, and I'm going to talk about, hopefully that you've done at least 1 semester of biochemistry, but you can see we characterized antibodies in 1959, and at that time we were struggling. We had some enzymes at that time, papain and pepsin. That's what you normally do. Remember, we eat protein, and there is a pepsin enzyme that's going to break the protein, proteolytic proteins. Now, as I said earlier, since this is a very important immune response in our body production of antibodies, so we have that controlled by our genes. So our genes control it. And I just told you, number 1, micro, delta, gamma, alpha, and epsilon. 1, 2, 3, 4, 5. So you have to remember 5 names. As I promised, I'm going to give you a mnemonic as well, and we will go from there. Let's look at some of the figures in the book, 4.2, 4.3. So this is a typical structure of immunoglobulin. It's just like me and you standing erect with 2 legs, 2 arms, and if you raise your arms like that, you basically are an immunoglobulin, right? So It has a heavy chain, so there is a heavy chain here, and there are light chains as well. And you can see, since this is a bigger molecule over here, is heavy as compared to lighter over here. And there are 1, 2, 3, 4 different components of this globular protein, and they are all joined together by disulfide bonds. And you can see from here, you can see double S bonds over here. Okay, disulfide bonds. And when we have two enzymes like papain or pepsin, if you use papain, this is going to cut over here. So what will happen is, if it cuts over here, so it's going to make an antibody divide into 3 parts. So 2 arms separately and the 2 legs together. So if you look at the structure, you will see these components. But if you were to use pepsin, it's going to break into 2. So this is important for some of the diagnostic ability that we will discuss in a while. Now, if you were to look at schematic representation of the whole molecule, you can see it's a globular protein, and you will see over here 2 important components of antibody. 2 important, and one is called constant fragment. So this one over here is a constant fragment. And the other fragment is called fragment for antigen binding. So just like myself, if I'm an antibody and I want to grab onto an antigen, so obviously I'm going to use my hands. So my hands over here, which are distal, are basically fragment for antigen binding. So I've got these fragments of antibinding over here. And you can see antigen binding. And then again, pay attention to the terminal. We have an amino terminal and carboxy terminal. And you can see over here that the amino terminal is outside and the carboxy terminal is inside. And then again, you will see the globular proteins over here, they also being united by disulfide bonds. So these are important aspects of the structures of antibody. The other important thing that I want to give you an upshot on that is that there is a constant portion, as we discussed over here, and these are carbohydrate moieties. So they are like proteins, and there are carbohydrates moieties. So these are like opsonizing things, so kind of sticky things. When you say carbohydrate, you talk about something that sticks. So you can see that there are CHO moieties attached over here. Right? And once we discuss complement fixation, when complement comes and fixes to something, so these are going to be docking sites for antibodies. And then again, the other important thing I want you to pay attention over here is that there are hinges. For example, if you know anatomy, there are hinge joints. So pelvis is a hinge joint, so you have pelvic joint right and left, right? And you have shoulder joints, because that gives you a limitation of movement. So you can kind of, it's a ball and socket joint over here and over the hips as well. So you have more liberty in terms of rotation in 360 for even dancers and 360 for gymnasts. You can see they can do that. But human beings do have a capability to do that. So that's called a hinge. Okay? The other important thing is that you have a fragment over here, that part of it is a fragment for antigen binding and fragment for constant portion that happens to be constant. So there is no variability there as compared to the distal portion shown over here in the color, like orange color. You will see that this is designated by V. So V comes from variable. I usually give this example. If I'm an antibody and I'm working in the lab, I want to hold on to something. So what I would do is, in order to protect myself, I'll always put the gloves on. So those gloves, there could be many different disposable gloves that I have. So in this case, what happens is that when I want to hold on to an antigen, I have that glove portion, basically, which is a variable portion. Okay? And also keep in mind that when I have these two arms and antigen binding, so there is a limit to what I can hold. So I can hold this like this. I can hold my computer. I can grasp this, but cannot lift it up. But again is that if I want to hold that, my arms would not stretch that much to hold a bigger molecule. You remember we talked about the sizes of molecules? So there is a limitation, physical limitation in terms of what is it that binds to antibody. Okay? Now, there are different amino acids. You see these terminal, N-terminal over here. They are sequences of proteins, and there are different amino acids, and we run them. We run them to find out what are the amino acids over here. And you can see that there is a variability of amino acid representing N-terminal. So we're just talking about the N-terminal. Now all of us, like 100 of us are here, we all have legs and arms. So we may have anatomically similarity. So all of us will have femur, tibia, fibula, humerus, radius, ulna, and so on and so forth, and even the muscles would be the same. But what do you think will differentiate us from each other in terms of anatomy? If antibody was like 2 legs and 2 arms, and these are something that we have common, so what do you think would differentiate us? If I say that we are all the same, but still we are very specific in terms of differences. What is it that you think anatomically would make each one of us unique? Louder? No. Not, what? Sorry? Appearance. Okay. Anatomically. Size and shapes, okay. More important than that, what would make you different than her? Sorry? DNA, of course. What else? I said anatomically, not genetically. Well, genetically as well, but think of simple things. Fingerprints. Fingerprints, right? We all have the same thing, same fingers, everything, but if you were to go the fingerprints, you'll be unique. Exactly the same way antibodies look the same, but when you want to do their fingerprints on the Fab portion, they are totally different and they are totally unique. There is no report as of today where they would come up with this idea that you share your fingerprints, right? So exactly, and this is the term that we use, fingerprints of these antibodies. Now, remember, if this happens to be an antigen, let's take this, platform as an antigen epitope, and there are many. It's a big molecule, and you know that the antibodies are relatively smaller, and they need to come and attach to it. So what will happen is, since they have limitation of a physical capability to hold on to that, so what they would do is, and you can see from here, light chain and heavy chain. If you saw, I did give an example for 2 legs and 2 arms of antibodies as being heavy chain. But if you look at antibody, I have to have at least a plaster or something else attached over here, which will be a lighter portion. And by the end of the day, I will have a lighter portion and heavy portion. So I'm going to grab onto an antigen with 1 amino acid of heavy portion, another amino acid for lighter portion, right? And then again, amino acids are like my fingers. So again, I can possibly stretch my finger to a certain limit, and I'm going to grab onto that. That exactly, the antibodies come over here, they want to grab onto an antigen. So what happens is, the idea is that more than one antibody can attach to an epitope, to an antigen. And if, for example, I can attach over there, I can lift it, but if I need to really move it, I'm going to ask at least 3 of you or 4 of you to come and help me out to move it, right? Exactly the same way, there's a sequence over here which is unique, and it is basically coming from CDR region, I'll explain that to you, where it actually sees the exact amino acid sequence and goes and binds that. So there is a amino acid residue in each and every heavy or light chain, which is going to go and dock onto an epitope. That's where the specificity is coming from. Now, the other important thing is about specificity is like endocrinology, where you have receptor and hormone. Antigen-antibody reaction, which is the basis of diagnostic test immune system, is also very specific, is a lock and key mechanism. So if you have a lock, so it should snugly fit, the key should snugly fit into that, right? And you can see there are serrations on the sides over here. They all look alike, right? But there's a number over there as an official key. And those of you who see the official key, it says over here, do not duplicate, right? So this means specificity. I'm going to go and open it up with this one. But physically remember that if this is an antigen that needs to bind to a antibody, right? So there are some limitation. In this case, you can see from here, if this is a perfect fit in for an antigen number 2 that binds with antibody number 1, but there are some chances that this can also come in and try to fit into the slot, right? So what I'm trying to say is that they all look alike, and if you go to my office, you can try this one. They may all go in, but would not open it. Does that make sense? So what happens is that sometimes an antigen-antibody snugly fits, but it is not going to initiate or mount an immune response if the physical connection doesn't exist. So that's where a physical connection has to come into place for a specific reaction like an antigen-antibody complex. Okay. Now, as I said earlier, I'm going to give you 3 terms: isotypes, allotypes, and idiotypes. And I'll explain that to you when I say 5 different types of antibody: gamma, alpha, micro, delta, and epsilon. So these are all different isotypes of an antibody. And within one isotype, if I tell you that there is a IgA1 and IgA2, right? So that will be basically allotype. Or if I tell you there's IgG1, IgG2, within one respective group, if you have more than one, depending upon the heavy and light chains, we call allotype. And idiotype will be antibody of human versus antibody of mice. And they are also important. Because what happens is, for example, if we want to produce a vaccine, so what we will do, we identify an antigen. And of course, we can put antigen in human being, because if you put an antigen in a human beings will respond in terms of antibody production. We'll take this antibody out, purify it, and give it to you as a vaccine. We cannot do it commercially because of the limitations. So what we would do is, and we can't even use a mice or rat because it's smaller animals. So we may want to use larger animals like horses. So you will take that antigen, give it to your horse, and the horse will produce tons of antibody. We take the blood out and purify it. But the question will come that is an idiotype. Will the horse antibody react and benefit you? So that will be a challenge for vaccine formation. Okay, sorry for this chart, and I think those of you who have books can better see it. I have some other, hopefully, better slide that are coming in. So this is a very good chart. I'm going to spend at least 10 minutes on that. And if you know this chart, you're done for today. Exactly everything. There was a time that we used to ask students to make a table, make pictures, but no, good thing is that it's been done for you. It's worth spending your money on. Okay? So what you see over here is, these are important features of immunoglobulin isotype. So they are, on left-hand column, there's a feature. I told you IgG, AMDE. They have molecular weights. They have additional protein subunits. And then they have a normal concentration in your blood. So if I was to run an antibody profile on you, I should see a normal antibody there, because they are in terms of milligram per mil. And then again, there is a total percentage of antibody, and then they are distributed in our body. Remember I said that they are either secreted or attached to the cell. Right? So there are antibodies everywhere in your body because they are protective. God forbid, if you don't have antibodies, that is a serious clinical disorder. Very serious clinical disorder. Then these antibodies basically have half-life. So what it means is, for example, if you give you an antibody, an antigen, and your body makes an antibody, that antibody will stay in your body for X amount of time and then removed. So there is a half-life. And then there are many different important physiological things that we need to know. For example, some of the antibodies in the intrauterine life can pass from mother to fetus. That's important for us to know, right? And some of the antibodies are basically present in milk. Some of the antibodies have this capability to activate complement. And then there are very specific antibodies. Remember about the other day when Dr. Coleman was asking you that you cannot attend the class if you are not updated with vaccination profile. So that vaccination profile is that, for example, if you tell me that I do not remember that I was ever given a hepatitis B, for some reason, I mean this is very unusual. So what they will do is that they will take your blood and look for those specific hepatitis B antibodies, and there's a titer there. If you don't remember, I'm coming from such and such country, I don't know whether my mom took me to physician, to pediatrician to get me vaccination, but we can run a profile on your serum and tell you exactly that you have a polio antibody, you have a DTT, you have an MMR, you have whatever. So this means that there are specific characteristics of these antibodies, right? Some of them basically are antiviral, some of them are antibacterial, some of them are antitoxin. For example, you get an emergency ER contact, Florida, I've just been bitten by a snake. So what do? You take it to ER. So the toxin is there. You want to neutralize a toxin. So guess what you're going to give it to the patient? Antibodies. Now the question is, as a physician, they will say, the nurse will say, okay, I'm going to give antibody. Which one you want to give it? Want me to give? You want to give G, A, M, D, or E? So I will say I would give G, because G has capability of being an antitoxin. You come to me and say, doctor, I have allergies. I think I may have inherited from mother because I get allergy to everything. So these are your claims in your words, and I want to demonstrate that. So I will run a profile on your blood, look for your antibodies, and guess what? Which antibody should I be looking for? E. Right? So you can see from here that each and every antibody is very specific. And we need to know for good, and we need to remember that. So you need to remember what are the sizes, everything over there. In short, all these things. And that's what, those of you who came late, I said that it's going to be a paired 1 point extra credit for each group to hand over to me by the end of the day. uh, an assignment that you come up with a mnemonic, with an activity that will help people to remember this information giving on this table, right? So that it kind of stays in your memory for a long time. We want to know their molecular weight. We want to know each and everything. And at least you can see there are 10 questions there. So there are 10 questions out there which you can guess. Okay? All right, let's move on. Now, you can see from here, IgG has 4 different types. Within one antibody, there could be very specific properties of those antibodies. So these antibodies basically, because we want to know, if I say protective antibodies, I want to find out if I give it to the mother and she's pregnant, would that antibody be transferred to the baby? If you remember me saying, I said all babies are sterile, right? But what if mother gets infected? If mother has HIV, so do you think that the baby in the intrauterine is going to get infected? Well, the question is we need to ask, can AIDS virus pass placenta? Can antibody pass placenta? So these are the questions you have to have. So in this case, you can see IgG basically has a placental passage. And that passage is not exactly by you saying it. When I say passage, this means a receptor there. And as a student, you need to know what is the mechanism, because we are going to devise some drugs. And the other important thing is that, remember I told you that either antibodies are free, they're floating around, or they are attached to the cells. So if there are 2 portions of antibodies, which one is called Fab, other is called FC. So which portion do you think will be used by these antibodies to be attached to cells? FC. I mean, I usually give an example. You stand on your legs. So you stand on the cell by FC portion, which are your legs, and Fabs are there. They attach, but they attach to antigens, immunogens. Okay, so this is an important concept that you have to keep in mind when you talk of these antibodies. Now, again, you will see over here, this is not from your book. The important thing in this slide is, if you pay attention, so you can see different types, we talk about that, and then again subtype, chains, and serum-like, and then they have a structure. So each and every antibody has a very definitive structure, and we need to know that. I'm going to spend a lot of time on that. But the important thing is that they have a function. For example, if I was to take your saliva and look for antibody, which antibody should I be looking for? A. Remember, at the moment when I ask you a question, just pay attention to the slide. The answer is there. If I was to take your B cell, I should say a patient's B cell, and then I want to find out if the patient has a B cell cancer, which antibody should I be looking for on the surface of B cell of that patient? B. Right? And if, for example, very common in this part of the world, all over the world, that round worm infestations with this like kids, and they have infestation of roundworms. I'm going to do their blood profile. So I would look for IgE, because body produces IgE as a defense mechanism against helminthic and even immediate hypersensitivity. That's an allergen. Right? And then I want to find out the other mechanisms for helping. Remember, antibodies can do the job on their own. They can restrict physically the invader. At the same time, the antibodies can also call for help. So you see a stranger, open the door, stranger wants to come in. Well, you can physically go and immobilize the invader, but you would rather call 911 as well for help. So antibodies will also call for help, and the help basically, of course, is going to come from larger. In the Incredible movie, it's going to come from Dad, right? So he's a big structure over there is going to come and help you out. And then again, if I want to find out whether you have a recent flu infection, for example, if God forbid somebody has a swine flu, I want to find out, do a blood analysis. Which antibody do you think is expected in the blood of a recently infected or flu person? G is there, but M. M basically is the one that is there for recent infection. G is already there. There are tons of G in your body, right? But you can see from here the structural and physiological concept of that. Okay? And then again, as I promised, I'm going to give you a mnemonic. So you cannot have the same mnemonic. Don't tell me that you made it. So copyright. So copyright mnemonic for remembering that table that I gave you as an assignment. And can I have a attendance sheet pass on as well so I know who's here today for me to accept the paired group? Just pass a sheet for attendance, please. Okay. There you go. So this is a mnemonic. Okay, interestingly, let's see. So I'm a pediatrician. Basically, I'm an immunologist, but I'm just posing as a. So I have one of my records, a family living in lymph node. And I've got a chart of this family, and I call it GAMD family. A GAMD family. Right? And the GAMD family basically comes that this family has 5 sisters. So it's like a sisters of 5 GAMD family, and you guess what their names are, simple. Gamma, alpha, micro, delta, and epsilon. And this is a mnemonic to remember the names of antibody, GAMD. Now, remember that I want to know their ages. So basically, so you can see that you say age multiplied by 10 just to make you remember that. And you can see over here, gamma is the oldest of the sister. And then there's an alpha, then there's a micro, there's a delta, and we have a new addition to the family, epsilon, which is like a newborn. Right? So what it would suggest to you that if you were to look at their serum levels, you will find them in that order. The highest serum level or the most half-life will be according to this chart because they are older antibodies, mature antibodies afterwards. Then, well, it's going to come as a recorded to you anyway. Yeah, don't worry. If you look at their date of birth, so date of birth will kind of give you a mnemonic for half-life. So 23rd of the month, and we have 2 sisters born on 5th of the month. One basically, then we had to say one was born in the morning, one was born in the evening, and we have delta born on 28th, and then we have epsilon born on 20th. And you can relate that to the chart, and we'll give you exactly what's happening. Well, mother should have done a better job of weaning them off from milk, but sometimes it happens. So she basically is an 8-year-old, or even, but she's still on milk. 1.3, 1 year and 3 months or 6 months, that's fine. They are still on the milk. But the other 2, though they are younger, they are not on milk. So they are not given milk as a food. And this mnemonic comes from the antibodies present in milk. For example, if you were to say mother has to breastfeed her baby, so what you're trying to say is that mother is giving gamma, alpha, and micro antibodies to the baby, and not so mother cannot give allergy by feeding milk by breastfeeding because it cannot pass. All right, and then again, remember that I'll have this record for Gambi family as well, and you can see from here that these sisters, they bond to their relatives. So if it's a lymph node like a home, mom and dad and kids and uncles and aunts and so on and so forth, there's a lot of activity going on. But since Gamma is the oldest of the sister, so she basically recognizes her uncles and aunts. Because all other sisters are too young to basically recognize all the uncles and aunts that are going to come and visit you in your home. And then again, whether these sisters are homebound or they go out, that's an analogy whether they are intravascular or extravascular. So if they stay at home, they are intravascular within the blood. But if they come out of the blood into joints, secretions, and other places, there's going to be extravascular. And simple for you to remember that this one being older, she can go on her own out of the home, but the rest of them pretty much are homebound. The other thing they noticed was that the oldest of the sister, the older was the sister, because when she was born, she was all by herself. So she had no sisters to play with. So as the family grew, so 1 from 2 to 3, so that kind of a ability to get attached with your siblings, so that sibling kind of activity increased. So the younger the sister is, the more capability of them for playing together, because that will help you to understand that the younger antibodies kind of have more attachment to each other, and they will go and look at the antigen. Well, only one goes to the school, which is G. So school vaccination, she's been vaccinated for all those vaccinations which are antibacterial, antivirus, antitoxin, and others are not. So this will give an analogy that only IgG is given as a vaccine when you have to fight bacteria, viruses, or toxins. No other. I did say the first antibody to appear is IgM, but I did not say when you have to vaccine, you have to give only IgG. Then again, if you look at the sisters' body shape, and you can see 3 of them are quite skinny. Alpha is kind of chubby, and micro is a really fatty one. Though it says micro, but unfortunately she's a little bit chubby. But if you look at antibody, micro antibody, the micro antibody basically has a pentamer. So this means if I'm a skinny person and I'm one antibody like G, and I want to compare myself with M, so 5 of me. So it's that kind of fatty, because it acts like a typical structure is like a, what do you call, a snowflake. And then if you look at their history, delivery, only micro was born as a C-section, right, and all or normal birth. And then again, as the younger, the sisters, because they're too young, they basically get hugs and kisses by their parents. So what it means is that delta is something that everybody wants to hold on to. So delta cannot exist on her own. She always has to be carried by mom and dad. So what I'm trying to imply is, if I ask you which antibody attaches to the cells, like T cells or B cells, which is mom and dad in this case, will be D. Because this is the younger antibody and you carry it. And last slide again is a recap of what I said earlier. A larger family, we already talked about B and T dad. Then we have other members in the family like macrophage, which is a big gluten, We have newts, which is a trash collector and disinfector, and there are some other natural killers or uncles. Then we have other families like ants, which are basophils, eosinophil. Mast cell, this is one of the ants of the family, and she's a pharmacist. Interesting. We'll discuss her in detail when we go for mast cell. Then we have a dendritic cell, that is an FBI agent. So one of the family member is an uncle, and it works as an FBI agent. Then we have Gamma sisters. Well, they have brothers as well. We call T brothers, and their names are helpers and regs, and they are CD4, and CD8 are the killers. And then they are family traits. We already discussed that because they are very specific. It's like a conservative family. Nobody's going to go out. They will only hang around with people that they know. So they are very restrictive, very conservative, right? And that basically is a part of the Gamma family where you really, really need to be very specific and move from there. And the last mnemonic for their biological activity is pain. And pain, basically P for precipitation, A for agglutination, I for immobilization, and N for neutralization. So these are some of the abilities of the basis of antibody and the tests that we will discuss and you will do in the class. So I'll stop here, and I think I haven't finished yet, so it's going to be a little bit an overlap. So we will continue.